What's good, y'all, and welcome back to another edition of Mr. Fitzgerald's biology class. Today we're diving into Unit 2 as a review of cells. Now this is a very broad unit, so we're going to kind of highlight some of the more important features or the older features that you just kind of need to quick grab. I'm going to let you know I'm not going to go into cell organelles on this. Those are things that you can quickly look up on your own if you need to, but these are more of the bigger concept things. So let's go ahead and get right into it. So theory and law. Remember, a theory is any sort of scientific explanation that's using data and multiple observations to come up with the best description of how or why something is happening. Okay? So it's not something that's made up, it's not something that's unproven, it's proven in the fact that it can make good predictions, it is supported by solid data and evidence, so all those pieces are there and it's our best inference of that data, meaning our best explanation of that data. Whereas a law is not necessarily more true or anything like that. A law is just a short mathematical, typically, description of something, or a short little phrase about something that's going to happen in nature that can also be used for predictions, like what goes up must come down. Very short to the point, not explaining how or why that's happening, it's just telling us that it's going to happen. And the biggest part out of these, and why I wanted to go over them, is to remind you that a law cannot become a theory and a theory cannot become a law. Just because a theory has good evidence and is supported does not mean it becomes a scientific law. Those are two separate things, right? Just like a long book with all its pages and words and explanations and details cannot become a single sentence, right? We can paraphrase it down to that, but we can't make it a single sentence, all right? So, the cell theory, why I wanted to go over that. Remember, the cell theory is that all living organisms are made of one or more cells, that cells are the foundation of all living things, and that cells come from pre-existing cells. So you can see that I have some thought questions here that were from an older PowerPoint. I just borrowed some slides because, hey, it's review, let's get it all together, right? But all living things are made of cells. Cells are the basic unit of all life, and that all living things come from other cells. All right. Now, let's get into the parts of the cell, specifically the cell membrane. So let's start with the outside and work our way in. So the membrane, remember, is a protective boundary. It kind of controls what kind of comes in and out of the cells. It allows certain materials in, it keeps certain materials out. And this allows the cell to have what's called homeostasis, or balance within the cell, allowing certain things in that are good for the cell and getting the waste products out of the cell as well. All right. Now, the cell membrane is also known as semi-permeable. What I was just kind of saying, allowing certain things in and keeping certain things out. Things that it allows in are usually very small things like oxygen and water. These are very small items that can kind of squeeze in between the gaps in between the membrane. Carbon dioxide is one that can go out. That's why I said it earlier. So that allows free flow of those very small, usually neutrally charged things to kind of flow between the cell membrane so that cell can be balanced. They can have that homeostasis. And how that semi-permeability works, or sometimes called selectively permeable, works is it's because this bilayer, these phospholipids here form a bilayer where their tails that are hydrophobic, the parts that hate water, are touching, and the heads, the round parts, are outside on the more of the aquatic environment. Sometimes they call it aqueous aquatic, meaning water. So I have all those heads that like water out here, and so basically items that are big or that are charged are not able to get through there. The big items kind of get bounced off because they're too large, but charged items, things that have large positives or negatives like sodium ions or chlorine ions from salt being dissolved in water, aren't able to fit through because these hydrophobic regions right here in the middle, these tails push those charged particles away because they don't like them, all right? So that allows it to be selectively permeable. Now this allows the idea of diffusion to work within the cell. Diffusion, remember, is all about moving from an area of high concentration to an area of low concentration. And when we have it all balanced, we have what's called a dynamic equilibrium. So the goal of diffusion is to get things that are very spread out very together to spread themselves out. So if I started off with a lot of particles here on the left side, they're going to move across the membrane until they're about equal on each side. And the reason that we call it a dynamic equilibrium is because particles will continue to move across the membrane, but their goal is to stay equal in concentration or amount on both sides. So overall, we would see no net movement, even though we might have one particle moving out. At the same time, we'll have one particle coming in to keep that balance. So the overall net does not change. Now, osmosis is the same thing. There's that diffusion move again. Because osmosis is just the diffusion move of water. So it's moving water from an area of high concentration to an area of low concentration. So things that are very condensed are high concentration, things that are spread out are low concentration. So because water moves from high to low, it tends to move in or out of the cell depending on where the least amount of water is. And that's going to depend on how much solute or stuff dissolved in water is in or out of the cell. And again, water can move across the cell membrane because of selective permeability. It's small enough to fit in between those gaps and get through without causing too much of a ruckus. All right, so there's three types of solutions that we have overall. We have hypotonic solutions, isotonic solutions, and hypertonic. And this is all about where the direction of water will flow with these solutes. The type of tonic solution, hypo, iso, or hyper, is telling you where the particles are in their majority. So a hypotonic, all right, when we're talking about the area outside, hypo means less. Hypo, that prefix means less. So I have less solutes, a less concentration of solutes, 1 dot out here, to my 6 dots that are inside the cell. So water here is going to actually flow into the cell. And the reason water is going to flow into the cell is if I have 1 dot out here, that means 1%. Let's imagine the dots as percentage. So 1% outside the cell is something dissolved in water. That means 99% is water. If I have 6% of things dissolved inside the cell, the other 94% is water. Well, 99 is greater than 94, right? So there's more water outside the cell than inside, so water will flow in. Let's take the opposite end of the spectrum and look at hyper before we go iso. All right, hyper means above. So if you look out here, there's a bunch of dots on the outside. There's actually 20 compared to the 6 on the inside of the cell. So if I have 20 dots in here, and we're going to pretend that the dots represent a percentage, it's easier to model this way. So I have 20% of stuff dissolved in water out here. That means 80% that remains is water. 6% of dots inside, that means 94% is water. So where is there more water? There's more water inside the cell than outside. So this is going to cause water to leak out of the cell, to move towards where there's less water, because there's more stuff over here, okay? So in this one, water will move into the cell, because there's more water outside than inside. In this one, there's more water inside the cell, so it moves out. That brings us to isotonic. Isotonic is equal. So if you look, there's 6 dots on the outside, 6 dots on the inside. Water will move, but it's going to move equally in and out of the cell at the exact same time because there's no spot that has more than another. So it's an equal movement across the membrane. All right, and it brings us into a passive transport, which we've kind of discussed a little bit with osmosis and diffusion. These things are things that occur naturally. You don't need any energy input to make them go. These molecules will naturally want to spread themselves out. So there's no energy required, hence that's why it's called passive, no energy. Okay? And these will occur through that selectively permeable membrane. Some good examples of this for the cell would be water. Osmosis will occur across this membrane without being stopped. Oxygen will move into the cell without being stopped. Usually the cell's using that oxygen to do cell respiration, so it's moving in pretty rapidly. And it's usually pushing out carbon dioxide pretty rapidly because there's so much carbon dioxide built up in the cell from cell respiration that it moves outward. So the cell doesn't have to do anything. These molecules move on their own based on where there's more of them. If there's more in one spot, it's going to spread to where there's less. Another type of passive transport is facilitated diffusion. So remember, diffusion is moving from high concentrations to low concentration. Well, facilitated diffusion works when you have things that are charged particles, like those sodium ions or chlorine items or potassium ions or different things around the cell, magnesium, calcium, etc., that need to get in that have charges. Or things that are big, like glucose molecules are actually kind of big. They're not able to fit through the little gaps, so they need some help. And the help here doesn't come in the form of energy moving them in or out of the cell. It just comes in these transport proteins, these channel proteins that are basically little holes or tubes for the solutes to move across. So let's pretend these are sugar molecules right here, these little circles, and these little particles can then move through these little tubes. There's a little tube there. It can move through and move its way into the cell because there's more sugar here than there is here. But it could be like a calcium or something else that needs to move into the cell. All right. Active transport, notice A, T, and P are in a different color there because it requires energy in the form of ATP. ATP is that energy currency for the cell. It's required to do work. Okay, so this is moving particles against where they want to go in areas of high concentration, or sorry, in areas of low concentration to areas of high concentration. So let's say I have this dot out here and it needs to move into the cell. Well, I can push that one into the cell. It doesn't want to come in because it's all condensed in here. There's similar particles in here, but we can force that in by using active transport to get that done. All right, so this is going to require some energy. It's going to require some proteins to get this one done. The proteins from passive transport with facilitated diffusion are more doorways. These ones are more of like pumps trying to force things to happen. And a great example of that is the sodium-potassium pump. So in this case, there's usually more sodium outside the cell than inside the cell. And there's more potassium inside the cell than outside the cell. The cell loves potassium. It wants to keep pulling more in. But it doesn't really like sodium. It wants to get rid of it. So it's moving the sodium against where it wants to go and the potassium against where it wants to go. And it used energy to get that going, okay? And that energy is in the form of ATP. So that brings us to prokaryotes. And prokaryotes are a type of cell overall. There's two types of cells. You have prokaryotic cells and eukaryotic cells. And what prokaryotic cells are, they're simple cells with no specialized structures. Their DNA is not contained in a nucleus. It's free floating in their cytoplasm. They really don't have too much. There is a cell membrane. You can kind of see this inner membrane here. They do have a cell wall for some. They might have a little tail-like extension called a flagellum, or little cilia, little hair-like extensions on the outside of them, but really nothing too special about them. Key components is they're usually smaller. Typically your bacteria cells, and they have to be unicellular. Because they don't have a lot of complex structure, they can't work cohesively to be a multicellular thing. They might chain up together, like this Staphylococcus aureus bacteria here makes these little chains, but that's just because they're anchoring to each other. They're not working together. It's not a single organism. It's individual organisms instead of one collective, okay? But again, their DNA is free floating in the cytoplasm. They have a membrane, probably a cell wall. Most bacteria have some sort of cell wall. And they do have these little specks in them, and those specks are going to be the ribosomes, and that's what actually does protein production. So that is a sort of specialized structure. As I know it says non-specialized structure, but a ribosome is kind of specialized in that it is there to generate proteins, because that's what the cell needs to survive. It needs proteins, all right? Eukaryotic cells are the much more complicated ones. And remember, karyote is Greek for nucleus or center. So in this case, these are those that have the nucleus, so the DNA is trapped inside there, and they also have different organelles. So like your endoplasmic reticulum, your Golgi apparatus, your mitochondria, chloroplast if you're a plant, lysosomes, all those different features are little organelles or specialized structures within the eukaryotic cell for it to do its job. These can be single-celled or multi-celled. An example would be like yeast. Yeast is a single-celled organism that we use to actually make our bread or beers or wines, and that's a single-celled fungus, and it actually breaks down some of those things through fermentation, which we talk about in another unit. Or they can be multicellular, like you or I, trees outside or different animals and plants. Those would be multicellular eukaryotes. There is another group in there called Protista. Protista are the ones that we couldn't really find a space for. They have a nucleus, they have specialized structures, but they might be like photosynthetic organisms, meaning they have chloroplast, but they also move, which is kind of strange. Amoebas fit in this category. So there's a lot of weird things within life that we haven't really figured out yet where they best fit, so we call them Protista. But also fungus is its own category as well, and they do have nucleuses just like we do, and they do have specialized structures as well. I'm not going to go into any organelles. You can look those up on your own, but hopefully this was a good kind of review for you on the Unit 2 stuff. That's all I have for today, and have a good one.